Now we can get on that EGR all the time.
Yeah.
This is where the light filters. I should so much. By a lot. Email. That's what's up? Good to see you again. Yeah, I know it's exciting. So is this a feminine lens via which to interpret brain injury, or is it brain injury to female?
Brain, so I put on ChatGPT and that's what I got. But I want to have like my view,
Yeah.
How to incorporate the female in your studies.
Yeah.
And
Actual.
what is a special for female, right? So nowadays, and I say, oh, you add a female in your study, people just dump female and then they say, oh, there is no sex difference. So now we will focus again on men.
Okay.
Right?
Well,
so
yeah.
But female has its own
Right.
but female has its own twist on it, right? Men don't go through pregnancy or migraine or menstrual cycle, and what changes might that impact the brain, and then you're looking. So you may not see at first sight sex difference, but there's actually a difference that you need to address.
Yeah, you for assays.
Yeah, so that's what I want to think.
Cool. How's the VA feel about that?
Yeah, it's okay.
They like it.
They have women's health portfolio.
Yeah, I know one of our paraplegic research participants is veteran. She goes to the VA all the time. Her name's Q.
Okay.
She's always like, there's no ladies over there. There's some.
So my husband grant got funded by VA for CPI in female on exactly the same.
Great.
But I'm good. Yeah, it's already 1 year old, but I'm going to talk about use of oral contraceptive and how that affects the brain. But I'm talking today mainly in terms of stroke. I will include one. Because I do have parallel data on PPI, but I haven't published, so I didn't want to put it. So I just put all the books. The thing is, some things are incomplete, but I can definitely show you when you're free.
Yeah, I appreciate that. My brother receives VA healthcare. He's in a RTMS protocol right now for major depressive disorder. But they do repeated transcranial magnetic stimulation of this area of the brain. So, yeah.
Which is really cool. So from that point of view, I'm bringing the whole body vibration.
Yeah, I've heard Helen talk about.
Yeah, so that's what I'm doing.
Oh, cool. Yeah, 7 stages.
Yeah. But again, I'm bringing all the published things, not unpublished yet, because unpublished never get like cleaned up till you put it for publication.
No, of course.
What parts? This is I did a something. Just project title description. Start. Right.
Okay. Well, if you want, I can give message you.
Yeah.
What I usually allow.
Sure.
Do they have content description? Because it used to have more sections with like training. Training program. Email? How do you?
Yeah, that's fine.
And then sort of the partner.
Yes. That's not.
Yes. What's that?
Thank you all for coming. You voted on me.
Yeah, so.
I think most people know. So thank you. Yeah. Yeah, so. We're
Percent.
That one.
Monday of this week, I posted the view so they can try it.
Yeah. Yeah.
The evening,
Yeah, so how
and
you
I
gonna
said,
do that?
oh, you are going to tell
How
me
you
that
gonna do
that's
that?
what it was.
Yeah.
But said, oh, you took on my brain now, I have to think, and I said, okay. But I said, everybody's thinking, but you are not actually thinking these factors, and then because that they are saying, and
Thought about special needs.
Yeah, I heard with feedback was good goes, so I've always heard that, sir.
Wait, this is what I haven't told you about that I've been really excited to tell you about.
Yeah.
This is a UN undergrad. She has scoliosis.
Yeah.
She was treated here. Started this nonprofit called Got Your Back Line.
Okay.
And she's in her first, second, but she's just starting, so she's like, her feet are a little.
Right.
I know I'm on the nonprofit board, I'm one of the founding members, and it's the scoliosis support group, so we talked about this one.
Yeah.
There isn't a local scoliosis learning why, there's been some challenges at startup, but she wants to make this an official UN undergrad.
Right.
So that would be, if you want to get in on it, that'd be, I've helped you out multiple boards, so I don't know how it goes. But yeah, I think that's the here. And then Dr. Joe Lai, I said that before.
The way you, I didn't see it, but I saw it before you got the third medical and.
And he is ortho, spine, and specialized scoliosis surgery, not the surgery, those would be your option. But comes in on the plan all the time. And it's pretty new here. She really likes me, so she is okay. Been a while, man. Been a while. Like 2 lifetimes. So excited. No, because we had it.
I know. I don't know, how it's gonna go on. I couldn't.
Like the folks. So far.
Yeah.
Have you talked to the?
No, I think it's.
I mean, you do live before.
Yeah.
And,
Yeah.
and you put it before, because I have a plan for one. You put yourself very small. Oh, do you want to sit on the edge? Yeah.
I don't.
Really? Yeah.
But that would be like a, I know that.
I can ask.
Neurologist that got fired from.
Oh, wow.
Yeah. Yeah, you'd rather just like, you shop around. Yeah, you wanna come back, I don't. Yeah, I would. Yeah. We could put up a few months. Right. Yeah, I can ask them. Yeah. You know, all set to make it known. Yeah. Right. Oh, yeah. I'm. Right, yeah. Really? I gotta think about that. Yeah. Right, right. People are gonna stop. They've been shaking that business.
That's the last, why I know I'm saying.
Yeah. Yeah. That's how class last time. Lost the tide.
I don't see anybody who doesn't know me, but by the way, I'm Ami Raval from neurology, and I can get started because I have 45 slides and I want to go through
till the last. So stroke disproportionately kills more women than men, if you don't know. And 1 in 5 women suffer stroke. Most strokes are ischemic stroke, and that's the focus of my research. Do I need mic or I'm okay? Mic's good? Okay. So there are typical risk factors for stroke that we all know. However, there are women-specific risk factors, and that's the focus of my research. So menstrual disorder, polycystic ovary, endometriosis, any impediments with fertility, migraine, oral contraceptive, complications of pregnancy, particularly preeclampsia, and menopause, and hormone replacement therapy. All this constitute risk factor for stroke, and not only risk factor, it also increases severity of stroke. So my today's talk is going to focus on 2 of the risk factors, that is oral contraceptive and menopause. I'll start with the oral contraceptive. Here you see there are different ways of contraceptive measures. Pills, commonly called oral contraceptive, they are used by 151 million users worldwide. The record is little older, 2019, but I checked new one, doesn't change much. One point I want to make on this slide is, even though it looks like only 16% of women use oral contraceptive, this number doesn't represent the real number. Why? Because in a developing country like where I come from, India, we have family planning centers, and in these family planning centers, oral contraceptives are kept on one desk. You can just pick up and walk away. And there are family centers, family planning centers who has volunteers. They go home to home in a poor neighborhood, and they actually deliver oral contraceptive because women might be working and can't go to the center. So they actually deliver in home. So there is no account for that. The other thing is, in United States or developing country, oral contraceptive is also prescription medicine for acne, or say endometrial problems or menstrual problems like dysmenorrhea. And this is prescribed and given to young girls early on when they have problems initiating their menstrual cycle. So this number that I'm bringing may not represent the true number. In United States, oral contraceptive was approved in 1960s, time of J.F. Kennedy, and this is a pharmacological drug used over 21 to 7 days, and it works by inhibiting hypothalamic pituitary ovarian axis. The association between oral contraceptive use and vascular complications was established since its approval in 1960. Within an year, there was a case that had pulmonary embolism and whatnot. So over the period of time, because of those early association between vascular complications and oral contraceptive, the formulation of oral contraceptive has been changed. And today's world, we have the 4th generation oral contraceptive. In 1960, the dose was 150 micrograms, and now it's 1/8 time less than what was initiated before. However, the dose that is today's oral contraceptive contains is much more higher than the physiologically circulating estrogen and progesterone in the system. So this overdose of hormones may be doing something. The older formulation had a high risk for ischemic stroke. Newer formulation has reduced risk of stroke, but however, newer papers are still showing that oral contraceptive increase risk and severity of stroke if one suffers stroke during the first year of their use. And this extra gonadal hormone may be complicating things because body is not used to having this extra ovarian hormones in the system, and that may be leading to this stroke early on. And if stroke happens, it may lead to more severity. The other interesting thing is there is still high risk if women combines this oral contraceptive use with the smoking. And that's what the question we wanted to ask. What happens in the brain, not in the blood? What happens in the brain if the combination is given? So we developed this rat model of oral contraceptive and nicotine exposure similar to that of women. So nicotine we give using osmotic pump, which is in those days when I developed the model in 2007 or 8, that time we mimic the moderate smoking paradigm. So 4.5 mg per kg per day is more or less, if one smokes moderately, they get this nicotine level. The oral contraceptive, we titrated based on the calorie consumption. So women has more or less 1,800 calorie per day compared to rat that is 300 plus grams, which requires 96 calorie. We give oral contraceptive for 3 days, and fourth day we give placebo. We allow this cycle to be repeated for 4 to 6, that is around 6 months worth of oral contraceptive use in women, or 12 to 15, that is more or less 1 year worth of oral contraceptive use in women. And at the end of this exposure, we expose animal to transient middle cerebral artery occlusion, allow them to survive for 21, 30 days, and do the histology. So here is the infarct volume quantification, and you can see in nicotine and oral contraceptive used exposed animal, there is increase in the infraction, particularly in adolescent as compared to adult. But however, in both adolescent and adult, there is increased infraction in nicotine plus oral contraceptive exposed animal. So the question comes, what could be the possible mechanism? So nicotine is an inhibitor of an enzyme called aromatase. Aromatase is an enzyme that converts testosterone into estradiol. And if women start using smoking early on in their life, and usually addiction starts at teenage, 16, 17, something like that. So those women who use nicotine early on in their life, they already have menstrual disturbance, which is very well known. And secondly, they also attain premature menopause. And remedy for disturbance in menstrual cycle or early menopause is oral contraceptive. So many countries gives oral contraceptive for treating this dysmenorrhea. So it goes like hand in hand without realizing what could be the complications. So as I said, we developed this model of oral contraceptive and nicotine use in the animal model. And then we wanted to see whether the estrogen level goes down or not. So this gray box represents the normal estradiol level during the normal estro cycle. And in case of nicotine plus oral contraceptive, the estrogen level came down. The other interesting thing is estrogen works via its receptor, alpha, beta, and G protein coupled receptor. So we were focusing on alpha and beta because these are the most potent estrogen receptors. What we found is in the estrogen receptor beta, particularly in the mitochondrial fraction, because now it is very well known that estrogen receptor is available away from nucleus, even it's present on the membrane, including mitochondrial membrane. And what we found is on the mitochondrial membrane fraction in a nicotine plus oral contraceptive treated animal, estrogen receptors were less in the hippocampus as well as in the cortex. So in a collaboration with Dr. Dave, we performed this very initial experiment sometime in 2010 or 12. And what we performed was, we isolated mitochondria from nicotine plus oral contraceptive, nicotine and saline treated animals from the hippocampus and cortex, and we monitored their mitochondrial complex enzyme activity. What we found is there was reduction in complex 4 enzyme activity. Other enzyme activity did not get affected. So we did a very simple experiment. We isolated mitochondria from the brain of a naive animal and then exposed them to ER alpha and ER beta agonist. And what we found is in ER beta exposed animal, there was increased activity. This gives us 2 ideas. One was nicotine toxicity is exacerbated in OC exposed animal while reducing ER beta mediated mitochondrial function. And the other thing is that ER beta regulates complex 4 enzyme activity. Collaboration with my late friend, Dr. Francisca Diaz, we performed a totally different experiment where we did autoradiography for nicotine and saline exposed animals. And here you can see in a nicotine exposed animal, there is a saturation of glucose. So if you pay attention to this grade here, you can see gray means 0 and 200 is almost at red. And you can see here more green saturation, suggesting that there is a glucose accumulation. Those who did autoradiography, they know this is the most tedious experiment and it takes long time to figure out what is changing. So me and Panchi decided to do metabolomics. We sent the sample, which was much easier to send it to company than doing this tedious autoradiography. So when the results came back, the first thing we looked for was whether the nicotine is reaching the brain. And here you can appreciate nicotine metabolites were present in both nicotine and nicotine plus oral contraceptive exposed animal. There was no significant difference between nicotine and nicotine plus oral contraceptive. But they were present, suggesting that whatever we are putting with osmotic pump is going to the brain. Most interestingly, what we found is nicotine and oral contraceptive alters the glycolytic metabolites, particularly those who are demarcated in a green are reduced, while the pyruvate demarcated in red was increased in the brain. This indicated the accumulation of pyruvate might have some impact on TCA cycle. We also found multiple metabolites of TCA cycle getting accumulated. So the first thing we did was to monitor what happens to the rate-limiting enzyme of glycolytic pathway. So we looked at hexokinase, and hexokinase is an enzyme that converts glucose into glucose 6-phosphate. And what we found is in an enzyme activity, in nicotine plus oral contraceptive group, there was a reduction in the enzyme activity of hexokinase, as well as there was reduction in the protein level of hexokinase via Western blotting. So the reduction in hexokinase activity could lead to problems with the glycolytic pathway, and hexokinase requires some post-translational modifications or allosteric regulations. However, given the low availability of protein, it may be just because of not enough of hexokinase in the system. We also monitor other enzymes of glycolytic pathway, and what we found is in most of this enzyme, nicotine plus oral contraceptive group had a low protein levels, particularly one that is LDH. And this increase in the pyruvate could be due to low availability of LDH enzyme, or this data also indicates that this may be trickling down to problems with the TCA cycle. When Shahil joined my lab as a research assistant, he was interested in further analyzing the metabolomics data. I'm not going to go in details of all the graphs here, but just wanted to bring up a cartoon showing that all the red indicated metabolites were accumulated while the ones in a green were reduced. So the main point I want to make from this busy slide is there was accumulation of phosphatidylcholine and there was accumulation of free fatty acids. One of the interesting thing of free fatty acid metabolism in the cell is that it's required for a process called palmitoylation. Palmitoylation is a post-translational modification, and this modification is required by multiple proteins in the cell for inserting themselves into the cell membrane. One of the proteins that we are interested in, estrogen receptor beta, as I showed you in the previous slide, that it was reduced because of nicotine plus oral contraceptive. So currently we put a grant that is looking at palmitoylation of estrogen receptor beta following nicotine plus oral contraceptive. You must have seen vape shop all around Miami. Every corner of Miami has a vape shop. So the idea is, in traditionally, when I developed a model in 2012, there was still traditional cigarette going on. But in 2010-12, you might have seen trickling down of electronic vaping shops in Miami. And within 3 or 4 years, every corner of Miami has a vape shop. So the idea was to develop electronic cigarette exposure model. So with the help of Dr. Zhao, first we developed a homemade unit and then ended up buying actually a unit that exposed rats to electronic cigarette vape. In collaboration with Dr. Zhao, we did some initial experiment, and figured out the cochitin level that mimics what is happening in human after electronic cigarette vaping. Vaping is a little more complicated than traditional cigarette because people don't have any control over vape. Like, we don't have any idea how much one vapes. So it's very complicated, but what we have found out is the cochitin level is more or less what is there with electronic cigarette vaping. So brief exposure to electronic cigarette vaping for almost 15-20 days shown the same energy metabolic disruption in the brain as we saw with the nicotine alone. And then we also found that electronic cigarette vaping exposure increased the infarct volume following stroke. It also worsened the post-cognitive uh, functions, particularly working memory. And we also found that the female rats were more susceptible to electronic cigarette vaping as compared to male rats, as far as the infarct volume is concerned. Subsequently, Vicente Sanchez, he joined my lab as a surf student and revisited my lab last year. He was working on some histological tissue, and he started training for immunohistochemistry of tryptophan and tyrosine hydroxylase positive neurons. And what he found is electronic cigarette exposure exacerbates ischemic loss of tyrosine and tryptophan hydroxylase positive neuron in the brain. And this is particularly interesting because people who suffer stroke, they not only have the cognitive decline, but they also have a depressive phenotype. And this may be showing that electronic cigarette may induce more depressive phenotype. So the question arises, so far I showed you more the combination nicotine and oral contraceptive, where I show you data on only nicotine. The question arises, what happens if you give oral contraceptive alone? And so as I mentioned, electronic cigarette is very newly introduced. So there is not much of a data that females suffer more stroke or severe stroke. That data is very flimsy, but there are data that showing cognitive decline and depressive behavior in women. And as I was talking, traditional cigarette, we always thought male is smoking more than women, but in electronic cigarette world, it's more enticing to female. The advertisement is more geared towards girls, particularly because they comes, if you have gone to any of the vape shop, the whole wall is full of variety of flavors, bubble flavor and whatnot. And I actually got interested in working on electronic cigarette because I teach undergrad, as many of you know. And one day I was finishing my class, and one of the girls forgot something looking like USB on her uh, desk. And I said, "Hey, you forgot your USB." The girl next to her started giggling, and I'm like, "Why are you laughing?" And she says, "You even don't know what is electronic cigarette." So electronic cigarette comes as little cartridge. You just put it on the back, which I now know. Jewel pods are little cartridge. And so I asked her, "Why are you smoke? Like, why are you using this?" And she says, "It reminds me when my dad used to take me to Disney World because it has same flavors of ice cream and candy." So that's what I said. This is something interesting, and I started exposing animals to electronic cigarette. I do have that what Dr. Zhao made. The homemade device is very funny, but yeah, I started like that. So I don't have a clear answer to your question, but I guess eventually we will know that. And that's why I wanted to test in a lab setting, because it will have earlier answer than human data. So oral contraceptive, Again, the same metabolomics data, we were just looking for what happens because of oral contraceptive. I didn't see a lot of changes just because of oral contraceptive. When Sabrina was in lab, she started looking each and every metabolite, what is changing in the oral contraceptive metabolomics data. We found that sphingosine 1-phosphate was increased, again, saturated, and sphingosine 1-phosphate had many targets in the brain. So how sphingosine 1-phosphate gets synthesized and broken down? It requires enzyme called sphingosine kinase 1 and 2. 2 is more prominent in brain as compared to other tissue, and it gets broken down by lyase. What we found in very, very pilot data, Indy was performing some Western blotting, and what she found is sphingosine kinase 2 protein level is increased while the lyase protein level is decreased. This may be a very initial hint that synthesis is increased, but the breakdown is not happening, but we need to confirm this. Sphingosine 1-phosphate had multiple receptors, at least we know 5 receptors. And again, Gina was doing early initial pilot data for one of our grants, and what she found is S1P receptor 3 is increased in Western blotting. S1P3 receptor is more on the microglia as compared to other cell types in the brain. And Sophie started quantifying microglial activation using shoal analysis that we learned from Roberta's lab. And what we found is there was a deramified microglia increase, suggesting maybe in oral contraceptive and MCO treated animals, there may be increased activation of microglia that could be the reason of increased ischemic damage. Again, these last few slides are very preliminary and we need to do more data. Yes, please.
Excuse me, what region of the brain are we looking at here?
So we are looking at the cortex, and that is a peri-infract cortex. So just to summarize what I said, I didn't show you all tons of data that we have published, but basically oral contraceptive and nicotine combination can affect aromatase enzyme activity, reduce estrogen, can affect the synaptic function. I also showed you data that it can have impact on the glucose metabolism, leading to mitochondrial dysfunction and maybe more neuronal cell death. What I didn't show you data today is about hypoperfusion. We have published a paper that shows nicotine plus oral contraceptive affects the histamine metabolism, and that may be route by which it may be reducing perfusion in the brain post-stroke. The other data that I did not show you is in a collaboration with Pablo Di Rivera-Vecchi, we performed this inflammasome study that is showing that combination increase the inflammasome activation, leading to maybe some defects in the endothelial dysfunction, leading to more blood-brain barrier compromise, leading to more severity of stroke. So this is more or less story about oral contraceptive. Moving to menopause. So yes. Worsening the effect because of the combination. The interesting part is what is getting worsened by what? That's what my question, and I'm still dealing with that, whether it's because of nicotine, because nicotine is a very dirty drug, as you know. It affects most of all the stuff. But what is typical of oral contraceptive? That's the question I'm still asking. Did I answer your question? This is more exciting part. So menopause, right? We don't talk about it. So it is very interesting when I go in a class and ask menopause, people start looking down. And the thing is, menopause is typical of human female. Apart from human female, only 5 species of killer whales, they experience menopause. And what I say menopause is a true menopause. menopause happens in many of the monkeys, but it's not true menopause because in most of the species where there is menstruation, when the menopause starts, animal's life is over. So in a human, we survive beyond our menopause, and that's what is called true menopause. When we talk about menopause, we always think it's ovarian thing, right? Oh, it's down there. No, it's actually brain thing. And that's what I want to emphasize because there are Shilohs, people who work on reproductive physiology, they work on menopause. And people who work in neurology, they say, oh, migraine or something associated or hot flashes, right? But they don't work together. So this thing is very sparse in our understanding, and we need a better understanding. So menopause is neurological. First hallmark of menopause, hot flashes or sleep disturbance, which I didn't realize till I started having this sleep disturbance. Mood change, cognitive fog, thermoregulation, instability, everything comes from the brain, right? The other interesting thing is estrogen is primarily neuroactive hormone, and estrogen in the brain declines before it declines in the ovary. So again, that is bringing that there is a neurological component bigger than ovarian component. And
Excuse me, when you say that estrogen declines first in brain
that's what the literature says.
as opposed to ovarian failure,
Yes.
do you mean the local production of estrogen?
Yes,
in the brain, or-
there is a de novo synthesis of estrogen in the brain, and that declines, and that only affects the hypothalamus and arcuate nucleus to stimulate ovaries to stop producing, stop developing follicular cells for the ovulation, and that can lead to increase in the FSH, reduce LH, and estrogen from the ovary.
So what you are indicating is that what is dropping is actually the peripheral aromatization, right?
In the brain, CNS aromatase.
Yes,
Yeah.
or in other tissues like
Yeah.
adipose tissue where estrogen is also
Yeah,
produced, or the
that's what
adrenal gland, or
mainly CNS, because a person might have little extra fat and they can still produce estrogen. That's a different story. But in the brain, CNS estrogen synthesis decline. De novo synthesis of estrogen in the brain declines. That's what. And that's what it affects the local neuron, hypothalamic neuron or arcuate nucleus neuron affecting the hypothalamic pituitary ovarian axis.
yeah, my point comes more about the ovarian failure, because if ovarian failure hasn't happened yet, the circulating estradiol produced by the ovaries, which is the largest by far, it's 99% of the of the estrogen is produced in the ovaries, right? It's going to into circulation.
I
And then
agree.
most of those estrogens target mainly the uterus, because that's the target organ mainly for estrogen. So I was wondering, there must be some kind of compensation since estrogens can close the BBB easily. So even if you have local, if the production diminishes in the brain, you still have the circulating levels. So I just
Exactly.
need to dissect apart,
Yeah.
how this can be detected before ovarian failure.
Excellent. So central nervous system or brain has de novo synthesis of estrogen. This de novo synthesis of estrogen regulates estrogen receptor on the local neurons, right? And the receptor of estrogen goes down. And that's why, even though in the system you have higher estrogen, the stimulation to this arcuate nucleus neurons is reduced. And that's why it affects the HP axis. That's what is the understanding from physiology of reproduction side. So again, the point I want to make is more neurological, and we need to pay attention because sometimes we just end up putting female in our studies, but we don't think about all those things. So again, this is the same diagram that I'm talking about. The most interesting thing here is normal reproductive cycle. You can see changes variation in FSH, LH, and inhibin during the normal cycle. But when the menopause hits, menopause is not a one day to another day thing. It takes years to set in, right? And that's what is called perimenopause, which takes almost 10 years. And this perimenopause is the most intriguing time of woman's life from various changes, as well as this is the time we need to understand better in order for us to prevent later on what happens. So bringing back to the stroke, women's risk of stroke exponentially increase after onset of menopause. And the interesting thing is, if the menopause happens prematurely before age of 42, 43, then that woman more likely to suffer stroke and severe stroke. We don't know why. And the other interesting thing is now women live almost 80 plus years. So 30 plus years of her life is under hypoestrogenic or postmenopausal, right? So this is another reason we need to understand menopause much better, because if we understand, we can reduce some of the comorbidities. Uh, Although I showed you a lot thing about menopause, the first drug came in market was in 2023. And this is the only drug available for menopause. So you can understand that people don't want to study menopause. And that's another reason that we need to emphasize study on understanding what leads to menopause, right? So this is the only drug that controls vasomotor effect. And what effect this drug has on a stroke outcome, we still don't know. So that could be another area to look it up. So how to mimic menopause in rat? This is a very interesting and tricky question because rat do not have menopause. I have read many of the papers that are published in a very high-end journal, and they say menopausal rat. Rat don't have menopause. So if you review a paper and if you see menopause rat or rat going through menopause, no. Because there is no menstruation, there is no menopause. That's the first thing. A rat we call astrocycle. Astros and menstruation are totally opposite word. Menstruation is either beginning of end of the cycle, while the astros is where the ovulation happens. That's the astrocycle, right? So there is a difference between astros and menstrual nomenclature. So people try to get confused in that. So that's one thing I want to bring it up. Astrocycle lacks menstruation, so no menopause. Traditionally, how we mimic menopause in rat is by performing ovarectomy. Ovarectomy in a human population happens. It's not never happens because women has to go through ovarectomy early on in their life because sometimes polycystic ovary or some cancer, stuff like that. So they get rid of ovaries. So surgical menopause does happen, but the surgical menopause is not typical menopause because there is abrupt loss of estrogen. The other model that recently Indy has been working on is called VCD model. And this is a chemical that affects ovary, leading to reduced follicles in the ovary, leading to slow transition of ovarian function as that happens in a menopause. And here this graph is showing you AMH, that is anti-Mullerian hormone. That is one of the indicator in rats. If that reduces, that means the menopause is setting up. We are also trying to do FSH because FSH should logically go up. But what model I want to create is of perimenopause, not postmenopausal. So I'm titrating the dose of VCD to figure out when the perimenopause we can mimic, how we can mimic the perimenopause. The other model that we routinely use in a rat is called intact ovarian age model, where we allow animal to, we buy retired breeder animals, which are 5-6 months old, allow them to be in a lab for a couple of more months and then monitor their estrous cycle over the period of time. Their estrous cycle become acyclic. They get stuck in one of the estrous cycle stage, maybe estrous or diestrous or metastrous. And then eventually they remain in a continuous diestrous where we monitor their hormone level. Estrogen goes down and follicle stimulating hormone comes up. That's what we consider it as a reproductively senescent female rats. So the mammals that don't go under menopause, they do undergo reproductive senescence. That means the age reproductive system, and that's what we use. Using this model of rats, we perform multiple experiments. One of the things that I'm going to bring up is post-stroke cognitive decline, because that's one of the biggest concern in the field of stroke. So in collaboration with my mentor and collaborator, Dr. Perez Pinzon, we perform these experiments where we are exposing animal to physical exercise of this reproductive senescent female and showing that physical exercise leads to improved cognitive outcome. There is a lot of literature also shows that physical exercise clinically or epidemiologically shown to improve the post-stroke cognition in stroke survivors. So in a collaboration with Dr. Helen Bramlett, usually she sits there, but she is not there. So we developed this model of low frequency whole body vibration. And why we develop is because menopausal women tends to get frail, particularly when the estrogen falls down, bone and muscles start weakening. So frailty is more common in female versus male. So if animals are frail, how can we put them to exercise? And we had a trouble when we used to put on a treadmill, some animals didn't perform good. So this could be a milder way of mimicking the exercise and what impact it might have on post-stroke cognition. So here you can see in the bottom, the rats are comfortable on this low vibrating platform, and this vibrating platform vibrates at 40 to 50 hertz. Your cell phone usually vibrates at 80 hertz if you have iPhone, and these vibrations are milder than that, so it's not causing any trouble. And the other interesting thing is on this platform, you can put animal on the next day. So we perform this low frequency whole body vibration. Yes.
Whole story associated with this experiment. I was reading a paper one day and found on South Beach, there were gyms where people were exercising on top of the vibration platform. It seemed to increase their muscle mass, things like that. So I went and talked to Adele Phil-Pate, who was downstairs at the time. And I said, have you ever heard of these vibration platforms? She goes, no, I've got one downstairs. Can I try it? So I got on it and my teeth were rattling. It wasn't the most pleasant thing in the world. So we got someone to build a chamber, and that's how this started, just based on reading the Miami Herald.
Why go to Adele?
Because it's a rehabilitation strategy for people with SCI. Yeah. We actually did a glucose metabolism study with rats that affected the vibration, but we have to publish it, but it seemed to have effect on neural tissue. Sorry.
That's fine. So there's a literature epidemiological that shows that whole body vibration also helps in human. Apart from that, there is this interesting study where it is well known that if stroke happens in a remote place, usually there is a procedure to follow that's called drip and ship. And so person will be given TPA and put on helicopter and then take it to stroke center. So the question was whether the helicopter vibration is actually helping or not. What they found is helicopter vibrations were not showing any neuroprotection, but low frequency vibration, they also had the same 40 hertz, and that was neuroprotective. So there is something outside of the field. This model of stroke is little different, but again, it gives us confidence that whole body vibration works. When Nadi was working with us as a postdoc with Dr. Bramlett and me, we performed this experiment where we exposed middle-aged rats to the stroke and followed by whole body vibration for 1 month, twice a day for 5 days a week. And what we found is whole body vibration reduced inflammation. But the interesting twist we got was one of the hormone that released from the muscle that's called irisin was increased in a whole body vibration group depicted here in a red bar. So we followed that irisin story, and what we did is we actually bought recombinant irisin and we treated animals following stroke, and Shahil was a lead person to do this experiment. And in RNA-seq data, what we found is there was increase or gene ontology suggested that there is an extracellular matrix and receptor signaling increased. So we followed what irisin is interacting with, And irisine is shown to interact with the integrin receptor alpha VB5. And here is the graph that is showing the genes for this two subunits are increased following irisine treatment. This is Shahil's story, so I'm not going to go in details because one of these days he can present all about his irisine data. So I will stop here, but what we are hypothesizing is the post-stroke irisine activates astrocytic integrin alpha VB5 signaling, particularly because this integrin alpha VB5 is located more on astrocyte. So currently we have AAV9 knockdown virus created, and he is knocking down in astrocyte, and we will see whether irisine still protects or not. So this is the experiment that he is doing, and one of this he will present. With this, I would like to thank all the members of my lab. I also like to thank Ophelia, Javier, Juliana, and Megan for all their help. Isabel from Dr. Perez Pinzon's lab, she is always instrumental in doing many of my experiments. Apart from that, all my collaborators, everyone, without their efforts and support, I cannot be here. And lastly, I like to thank Chantal Scheinberg. And once upon a time, I had a hard time. Even though Trump was not there, I had a very hard time, and she donated some money, and I could survive in this institute. And I am still thankful to her for all her well wishes and her support in women's health. So with this, I want to take your 40 seconds one more and show you this interesting video if I can, because that's what I want you to go home with.
Are you sure? There are a lot of advantages to buying in bulk. For example, I noticed that you purchase your tampons 1 month supply at a time. What? Think about it. It's a product that doesn't spoil, and you're going to be needing them for at least the next 30 years.
You want me to buy 30 years worth of tampons?
Well, 30, 35. When did your mother go into menopause?
Talking about this with you.
This is the reality. Nobody talks about it. So I guess we need to understand how these things work. Thank you. I mostly collaborate with many of the Miami project PIs, but in future, I want to do spatial transcriptomics. So somebody's looking in phone, maybe he can help me. And other person, I wanted to collaborate with Dr. Ali Hassan. He has a very cool compound that can be useful for, I'm blanking the name, TMP or TPM. Yeah, that's what I want to try in a stroke. It may be very cool, and he has a cool data on TBI. So that's what I'm looking forward. Apart from that, I'm using many of the instruments. Just now I mentioned that we are isolating astrocyte. Shahil uses Pablo's lab all the time to isolate astrocyte and many other core facility I use. Hopefully I don't have to pay for core facility, maybe being a part of Miami project. That is the idea, but whatever it is. Did I answer your question? Okay. So traditionally, what we know is before any disturbance in estrogen circulation, premenopausal women suffer less stroke, postmenopausal women suffer more stroke. That is traditional knowledge. But now what we are digging into more, and what we are realizing is there are women-specific risk factors that may put them like at high risk for stroke early on in their life. And if that is to be taken care of, then we need to have model or some understanding why they suffer stroke earlier in their life. Preeclampsia is one of the complications that women has. Only 10% of women has pregnancy related complications, but that 10% may likely to suffer more stroke, higher stroke risk or severe stroke, right? So the point I want to bring is because NIH mandate us to include females, we are including female in all our experiments, but then what we come to is, oh, male and female didn't have sex difference, so we throw female out and we just focus on male, but female has a whole lot of comorbidities that we need to target. Yes, I do have to include males because if I write on oral contraceptive, I don't. But if I am writing on menopause and if I am trying some drug, like I just mentioned that I want to use this new drug, if I am putting the drug, even though I am putting estrogen receptor beta agonist, I do have to put male rats because reviewers want to see what happens in male, right? So I don't get a free pass, but the people who work on male system, they get a free pass, not including female. Comorbidities. Lower. Yeah. Yeah. So the other thing I'll bring up, I brought menopause and I made it a big fuss about menopause, but women who goes through menopause, we don't know they were multiparous, nulliparous, right? Their complications are different. They had 2 kids, 5 kids, no kids, right? So if women didn't have kids, their risk for ovarian cancer is high. So stuff like that, we don't know, we didn't dissect. And one of the person, Dr. Louise McLaughlin, she works on women's health all the time with the stroke. And I asked her the same question and she says, this is a can of worm. If we start opening that, it's going to be very complicated. But we don't know who suffers more stroke because basically in a woman's life, because of the menstrual cycle, the clotting factor also has a cycle, right? And these clotting factors get affected by estrogen, then it may lead to more clot somewhere. That may lead to more stroke. So we don't know what complicates what. So there are a lot many questions to be asked, but the beginning could be at least include comorbidities. Like if you are having a rat model, maybe you are including female, maybe you want to say reproductively senescent female versus young female or something like that. Maybe slowly the data comes out. Thank you. So I did not do, but Dr. Bramlett's previous study on spinal cord had amplitude, and what I adapted is 40 hertz from her paper. So I myself has not done in a stroke, and definitely I can do. The person sitting next to you, I always ask for doing lactate shuttle. We never end up doing that. So we will have to do that. I agree with you. There are a lot many things in there. This was metabolomics, that is global metabolomics, and we don't know it's astrocyte, microglia, neuron, right? So those kind of things also need to be And that's why I'm interested in maybe going something like spatial things. Again, yeah, definitely.
So my question is related to the fact that women, postmenopausal women under hormone replacement therapy, do you know, to your knowledge, if there are differences in terms of the stroke as a risk factor, or in terms of percentage of women suffering from stroke? Are there?
Excellent question. So women's estrogen for stroke was used for clinical trial. There were at least 4 clinical trials where estrogen was given postmenopausally to reduce the risk of stroke or cardiovascular incidences. All the trials in late 90s or early 2000 were failed, and actually they had to be stopped prematurely because women who were on estrogen or hormone replacement therapy, they suffer more stroke or cardiovascular incidences. Why? Because most of these trials had women recruited, whether they were premenopausal, during their perimenopausal, postmenopausal, and years after menopause, right? So that was all over the place, and the data was not very clean. After that, working on estrogen was frowned upon. So if you look at the research papers, they were literally decline in the estrogen field. And then it got revived because more question came out. What happens if the estrogen start right at the perimenopause versus postmenopause? The other interesting thing is the estrogen is given in a continuous fashion, like 1 pill a day, but in a natural release, estrogen is released in a cyclic fashion. So this continuous may not be the most effective way of treating. So those are the questions that I personally like to answer, and I do some research on that, but there is not much in the field out.
Your research is very interesting, and I have another question, and I apologize for holding this meeting for so long, but so in your models, when you give estrogen or any other agonists, how can you set apart the estrogen signaling in the non-genomic signaling pathway versus the genomic, which is the one that takes longer, right? You have the rapid signaling, which is due to the estrogen binding to G protein coupling at the membrane, right? And this is especially relevant in neurons. And typically this, the triggering of this signaling is related to neuroprotection, right? And then you have the genomic, the classical one that goes directly to the receptors in the nuclear compartment and execute, gene transcription, taking longer.
Again, excellent question. So I'm not doing those experiments recently because I move on to estrogen receptor beta specifically, but there is a compound that estrogen is bound with a BSA, a bovine serum albumin, and that only can stimulate the membrane-bound receptor signaling, or in mitochondria, when we isolate and we put that BSA associated estrogen, then it only affects the surface receptor, not the one in the nucleus. So that's the way to dissect out. And I did some experiment in past, but I'm not doing that currently.
Thank you very much.
Welcome. So in my research, I do variety of animal models. As I was showing, I have 4 animal models of menopause kind of. So overectomy, I've done lot in past and I continuously do. I also do VCD, that model I showed you, chemical that affects the ovarian follicles, and that I can do it in a younger animal, because when I put my grant, one criticism always comes is when we use retired breeder or reproductively senescent animal, their age and they may not perform good on water maze or behavioral tasks. So I have to address that, and that's why I developed this VCD model. So that's what I'm trying to do.
Thank you.
Thank you. Thank you very much.